but I mean, like, I've never heard, you won't have an ejection fraction of 90 or 100. You won't have that. There'll always be a little bit of blood remaining in the ventricle. The only time I can see you having an ejection fraction that high is if you're taking certain medications, like if you're normal and you're taking digitalis, which actually is what we call a cardiac inotrope. It makes the heart muscles contract vigorously and firmly and strongly, and that can actually cause more to be ejected. So if you were normal, if everything about you was normal, your heart was normal and healthy, and you took digitalis, it would increase your ejection fraction. If you took certain types of drugs like cocaine, it can do that too. I don't know if amphetamines will increase your heart rate, but I don't know if it will increase the ejection fraction. It will increase the heart rate though. So, you know, as long as your heart rate isn't tachycardic, if your ejection fraction is high, it shouldn't be a problem. Okay. And let's move on. We'll be out of here soon. Just want to make sure that I covered all of this stuff here for you guys. Stroke volume. Did I answer your question? Okay. Alright. And it's very common. Like if somebody has an ejection fraction that's low, people who have, for example, congestive heart failure, what you hear is congestive heart failure, we'll talk about that another time, that actually will lead to a low ejection fraction. And that low ejection fraction tells you the person's heart is failing. It's not actually being effectively pumped. Okay? Okay. Now, when the heart pumps, it's going to push blood into blood vessels. And as that blood flows through the blood vessels, it flows under a certain amount of pressure. And when we do the circulatory system and look at the blood vessels, we'll see how the pressure changes every time blood goes through various types of vessels. But there's several things that can affect the pressure that blood is flowing under, or even the speed that blood flows. And we can see here that one of those things that can affect it is resistance, what we call resistance. And an example of that is if blood vessels narrow or if they get larger, that can give more space or less space for blood to flow through. Another thing is if somebody has clogged arteries, the actual collection of plaques lining blood vessels is disruptive to blood flow. It actually can disrupt blood flow. Also, if people have valves that are stenotic or prolapsed, it can actually cause resistance to the passage of blood through that structure. And here we could see that very often blood, as it flows through vessels, is under pressure and generally will flow from the area where there's higher pressure to lower pressure. Okay? And that certain things can affect that pressure. If you have problems that cause increase of friction inside the blood vessels, like, as I said, atherosclerosis and arteriosclerosis, pathologies that leave plaques of cholesterol and calcified plaques in the blood vessels, it actually gives, instead of a nice smooth vessel wall, you have a bumpy vessel wall. Sometimes it actually narrows the vessel wall. And these actually can cause a disruption in the blood pressure, the pressure that blood is flowing through. It usually makes the pressure elevate, makes the pressure go up. As blood vessels get narrow, it also makes blood pressure go up. And it's just like we saw with the heart. When the heart walls contract and constrict on the blood that's in there, the pressure inside goes up. Same thing with the blood vessels. If blood flowing through a blood vessel, and that blood vessel decides to constrict, get narrower, the pressure inside it is going to go up. That pressure can also be affected by any kind of plaques that build up on the vessel wall. Somebody has a really bad diet, they have high cholesterol, plaques are building up on the endothelium or under the endothelium. It narrows the blood vessel. The pressure inside that particular area is going to be high. And we can see here that this kind of resistance from plaques or problems with the vessel wall can be caused by vasoconstriction, which is actually constriction or narrowing of a blood vessel, atherosclerosis, which is a pathology where cholesterol plaques develop inside blood vessels, and hypertension. Hypertension refers to increased blood pressure, above normal blood pressure. But hypertension is directly a result of things such as vasoconstriction and atherosclerosis and some other problems. Just based upon what we've done in anatomy and physiology so far, can you guys tell me something that will cause hypertension? Something from the last section, one of the last sections we did.
High sodium.
High sodium will increase our blood volume, and you have more blood volume, you can also have a higher blood pressure because you have more volume inside the same space. So you increase the pressure. So sodium will do that. What else will do it? How about stress? What if you're under a lot of stress? Yes. What hormone will you release if you're under stress? You release what hormone? Anybody remember? Cortisol. And cortisol will cause vasoconstriction and hypertension.
Oh, yeah.
How much time do we have? We have like 12. Do you guys need to take a break? No. How many want to take a break? Raise your hand.
We'll take a break.
You want to finish? Let me see how much more I have. I have to do the characteristics of muscle tissue and electrocardiogram. We'll have to do, I guess, next week. We do electrocardiogram in lab, and then the overall control. All right, let's just go as far as we possibly can. All right. Is it really? Oh, yeah, I can see that. The sun is just...
Oh my gosh.
Yeah. Just wait till, yeah. All right. Where are we? All right, let's look at the muscle tissue itself, the myocardial tissue, okay? Because this is the introduction to the next thing. What actually controls that heartbeat? How is it that we have 70 beats per minute? What's causing that to be to happen? And we actually have nervous control. But it's important to look at the cardiac muscle tissue first, because our cardiac muscle tissue, even though it's muscle tissue, and if you remember from A&P1, it was striated, right? It has some very special characteristics. These muscle fibers, which you see here in the kind of bamboo formation, they've got many nuclei. They have these structures. Do you remember those structures here?
Intercalated.
Right, the intercalated discs. These are actually found at the end of one muscle fiber and the beginning of the next. And these intercalated discs are very important because they allow one fiber to communicate very well with the next fiber. And these fibers love to communicate. Now, let me tell you some things that are very special about cardiac muscle fibers, besides the fact that they're branching, besides the fact that they've got a single nucleus for each fiber, and besides the fact that they have intercalated discs. They are capable of being autorhythmic. That means that each individual muscle fiber is able to maintain its own rhythm. And in fact, they can communicate with each other and form something called a syncytium. A syncytium, let's see, where's that word? Here it is. Syncytium, S-Y-N-C-Y-T-I-U-S. Okay, what does that mean? Well, if I had somebody living in front of me and I opened up their chest and I saw their heart and it was their beating and I dissected out a couple of muscle fibers, say from the ventricle, and put them in a little petri dish and separated them from each other, I could look at those muscle fibers and be sitting there contracting on their own, at their own little rates, each of those muscle fibers. And if I brought them near each other, then suddenly, after a minute or two, they would all start contracting at the same rate, because they each are able to contract on their own without the nervous system telling them, okay, contract. They don't need, like our skeletal muscle, a signal from a motor neuron to say contract. They can do it on their own. They can develop their own action potential and be autorhythmic. And not only that, but they have their own idea of how many times they should contract every minute. In fact, all of the fibers of the ventricles and all the fibers of the atria form their own syncytium. The muscle fibers of the ventricles, they like to be at approximately 50 beats per minute. Okay, so if we took out just the fibers of the ventricles and put them in that dish and put them next to each other, they would communicate with each other and they would start doing their contractions at about 50 beats per minute. If I took out atrial fibers from the atria of this person, I put them in a dish and watched them contract and then communicate with each other, they would eventually agree to beat at a rate of about 120 beats per minute. So the syncytium of the atria is about 120 beats per minute, if left alone on their own. And the syncytium of the ventricles is about 50 beats per minute. So how does our heart work when it has two groups of fibers that have their own idea of how fast the heart should beat? Well, they are brought together in one organ, and they have overall control by a nervous system, a actual series of special fibers or special neurons that tell them what our heart rate should be. Okay. So here are the cardiac muscle characteristics: branched, intercalated discs. They can form a syncytium, and they are autorhythmic. Okay, and just remember the syncytium of the atria is approximately 120 beats per minute, and the syncytium of the ventricles tends to be about 50 beats per minute. Well, why is that important? Well, you know, it'll be important in people who have pathologies, people who have, for example, tachycardia, and they've been diagnosed with atrial tachycardia, and their heart rate is like 125 beats per minute or more. You know, it's because suddenly their atria have decided to revolt and just take over and tell everyone what their heart rate should be. And it's not the atria's job. So, all right, here we go, more muscle fibers, and let me just go. Okay, here we go. So we talked about the two syncytia of the heart. The atria themselves have one syncytium, about 125 beats per minute, and the ventricles a much slower syncytium. These two groups of muscle fibers are separated by a piece of connective tissue, and that's called a fibroseptum. And you can see it right here in yellow, dividing the atria from the ventricles. This fibroseptum develops during embryonic development, and it separated these two groups of autorhythmic myocardial fibers. Okay, so these two groups of fibers ended up developing their own syncytium, their own heart rate. Okay, now in order to, here's some more characteristics of myocardial fibers. We got our autorhythmicity, myogenicity. They can spontaneously depolarize, so they're autorhythmic, and they are contractile, and that means they contract. You guys know all that, right? Okay. Whatever. It's just everything I just said before. Okay. So how do we get the atria and the ventricles to work together and come up with some reasonable heart rate of about 70 beats per minute? Well, we have something called myogenic nodal cells. Myogenic nodal cells are specialized cells that are able to conduct electrical impulse across that fibroseptum. And the myogenic nodal cells decide what the heart rate is going to be. It tells the atrial syncytium, this is what our heart rate is. And it tells the ventricular syncytium, this is what our heart rate is. And that is what controls the actual cardiac cycle, how many beats per minute, and the contraction or the systolic events of the cardiac cycle. And I know here there's a graph here with things like fast calcium channels and potassium channels. I'm not going to go into this. This is the only three words that I want you to get from this particular slide. Myogenic nodal cells, okay? All right? Don't go crazy with all this other stuff, all right?